let's reset ourselves. We're going to kind of switch gears. So, on Wednesday, we were talking about cellular respiration. I kind of have to switch gears here. It took my brain a hot minute. We had started cellular respiration. Woo, it's taking me a real minute here. Okay, so we had done the first stage, which is glycolysis. Okay. Remember that first stage took us from glucose, a 6-carbon molecule, through a series of steps to pyruvate. A 3-carbon molecule. And so remember, for a 3-carbon molecule, and we need 2 of them. So we still have 6 carbons total. And count our carbons. All right, so we left off on Wednesday having just started the prep step, also called the link reaction. Remember, our overall goal here is to get to that citric acid cycle, because a lot happens in that citric acid cycle that's actually valuable. And as we can tell gleaning here, not a whole lot happens. Okay. So we want to think about answering those major questions. And we'd answered a few before we left class. Okay, and remember, where are we? On the mitochondria. And what was our goal? We'd started as pyruvate. Okay, and we wanted to end as acetyl-CoA. That a little bit. Remember, the key behind being acetyl-CoA is that CoA group. Right, we want to think about that being a little flag. And that little flag is going to be really useful when we get into the citric acid cycle, right? Because it's going to be able to now enter the citric acid cycle, and that flag is going to be able then to be recognized quite literally. Here I am, see me now. Use me now, in a way that pyruvate was not. Okay, so here's our goal. Acetyl-CoA. Excuse me. Okay, so we had just started talking then about our counting the carbons. So remember that pyruvate is a 1, 2, 3-carbon molecule. That acetyl-CoA is only a 2-carbon molecule. Okay, so we lost that carbon along the way. And we can't magic stuff away, right? This is one of our laws. Okay, so we had to have done something with it. And that something was decarboxylation. So we've lost carbon dioxide. We talked about decarboxylation when we did our recitation packet in lab. Don't forget about those recitation packets, right? They make really good study guides now that we're doing this again in lecture. Okay, what happens to that carbon dioxide? Do we just store it in the cell? Ventilate it out, right? So remember, this is our first connection between cell respiration and ventilation, right? We're going to breathe that right out. Okay. So one more important thing that we see when it comes to this carbon dioxide. It's another answer to one of our questions. Okay, in order to make carbon dioxide, okay, we have that carbon that we stole from our pyruvate, and I spy with my little eye oxygen. Okay, so we need oxygen in order to make that carbon dioxide. And so oxygen needs to be around for the prep step to occur. So sometimes when we think about oxygen, it's not as straightforward as it always appears to be. All right, last couple of questions. Do we have any ATP? No ATP. Do we have any other energetic molecules? We do, right? Got ourselves a coupon. All right. How many coupons did we make? 2. Perfect. Right. Remember, we started with 2 pyruvate. So we're going to go through this process once. Oops. Twice. Okay, so we're going to get 1 set acetyl-CoA and NADH, 2 sets acetyl-CoA NADH. Okay, so this is why it was so important that we really focused when we were doing glycolysis that we got 2 pyruvates, so we could hang on to the fact that we would then have 2 pyruvates when we got to the prep step. Right, because now we're going to have 2 acetyl-CoAs and 2 NADHs. Of course, we let off 2 of these too. We're not going to hang on to the carbon dioxide. Okay. So we're going to put the 2 NADHs in our piggy bank. Remember, we're saving up those coupons for later. Okay, but now I have 2 acetyl-CoAs to run into the citric acid cycle with. Any questions about anything that we've done in the prep step? Okay, so here we are, ready to move into stage 3 of cell respiration, which I would argue is the most complicated of the 5 stages. Okay. But you guys have also already been exposed to it before, right? We got this far in lab. So we still are on a little bit of familiar territory, which is nice. Okay. So we're still going to do a version of citric acid cycle, say that 5 times fast, light. Okay. And even though we're going to add some more details to it, okay, we still want to make sure that we don't get overwhelmed. Okay. We want to stay focused on our box. What's the goal? All right, so what do we go in with and what are we ending with? And it's a cycle, so that's not quite as straightforward as it has been in the past, but we can still focus on that. Okay. Where in the cell are we? Are we going to consume any kind of O2 or oxygen products? And are we going to get ATP or any kind of energy out of it? Okay. So we want to hang on to our goal box. All right, let's rock this. Okay, the citric acid cycle, which is also called the Krebs cycle, named after the gentleman that discovered it. I tend to call it the citric acid cycle because that's named after one of the compounds actually in it. I think it's a lot more useful. Also, I hate stuff that's named after people. Okay, so this is aerobic respiration here. Okay. And I'm pointing this out in advance because as we go through it, sometimes it feels overwhelming. So here's an answer to one of our questions. Okay, this is aerobic respiration for two reasons. The first of which already feels familiar. So we'll look at the second bullet point. Right, because again, we're going to produce some carbon dioxide. Okay, so once again, I spy with my little I, some oxygen. Right? You need oxygen to create carbon dioxide. Okay, something to bind with that carbon. Okay. We're also, as we're going to find, okay, use some products, okay, some larger compounds, okay, that need oxygen to exist. Okay, so we lightly kind of touched on this in lab. Okay, but it was really in the background. Okay, so we're going to look at this a little deeper. So in both cases here, okay, O2 is not super apparent, okay, but it is very important. Okay, and so the key here is the citric acid cycle is not going to continue to turn. It's not going to continue to run if oxygen is not present in the cell. So that's what we mean when we say aerobic respiration, right? Oxygen needs to be present, even if we can't see it in this case, or it's kind of hidden. So it's kind of like a Where's Waldo book. The other thing we're going to remember is that we're in the mitochondria here, right? Remember that we entered when we were in the prep step. And remember, when we enter the mitochondria, we never leave the mitochondria. It's like Alcatraz. Okay. So we're going to stay in the mitochondria for the rest of cell respiration. So we can feel comfortable knowing that the mitochondria is going to be the answer for the rest of our cell respiration location questions. All right, everybody ready? Here we are in the mitochondria. Okay. Here's our citric acid cycle. We can tell by the drawing that it's a cycle. Okay. And now we can see that for once, and from what feels like forever, okay, we're going to get a little ATP out of the dealio again. So let's start to look at some summary of what we should expect as we go into the citric acid cycle. Okay, so the citric acid cycle is quite large. It has 8 steps. I am not going to make you know them. Okay? Some advanced biology class, like cell and molecular, will. We are not in cell and molecular. So know that it has many steps. We're just going to focus on a few important ones. Okay, mostly step 1. Okay. So step 1 is where our acetyl-CoA comes in. The one that we worked so hard to make in the prep step, right? Okay, so remember, acetyl-CoA is a 2-carbon molecule. Put a flag on it. And it's joining up with a compound coming off of the last step. Remember, this is a cycle. And that compound's called oxaloacetate. I didn't name these things, man. Okay, that's a 6-carbon molecule. All right, so here's the deal. Oxaloacetate, right? Oxalo, okay? Oxygen, right? They sound the same. Okay? So that's the compound that makes this aerobic. So if we don't have oxygen floating around in the cell, we can't make oxaloacetate. Okay? We don't have oxaloacetate, we're missing a key piece of our citric acid cycle, right? That's one of the reasons that this is an aerobic process. Okay, acetyl-CoA plus oxaloacetate makes citrate. Okay. Citrate is what the citric acid cycle is named after. It's one of the compounds that makes your oranges taste so good. Okay. You're not going to need to know the structure of any of these things. Now, what's key is in the steps 2 through 8, in some very specific processes we are not going to stress about. Besides, we are going to use decarboxylation. We're going to lose some CO2. Okay. Twice, in fact, right? We're going to end up back at oxaloacetate. Okay. Along the way, as we break those bonds, we release some energy. So we're going to get some ATP. And we're going to get a whole bunch of these electron carriers. They are coupons. Okay. Well, the ATP is good. We know we like ATP. The big key behind the citric acid cycle is it pumps out a lot of coupons. Okay, way more than we've been getting at any stage before now. And remember, we've been talking about the key behind these coupons is they let us cheat at active transport. And the active transport is the step after this, after the citric acid cycle. So we're really going to ramp up our coupon pile. Okay, We're going to stop here. On Monday, we'll look at the cycle itself and then actually make some ATP. We'll probably finish cell respiration on Monday. Huzzah! Stop the recording, please.